Hey everybody, how are you doing? Good to see you. I've noticed that some of you I am seeing on the Google Hangout as well. I'm excited to talk to you there, and that's great. Hello, everyone. So, welcome. I'm excited to start a new unit with you. We did finish evolution right before the break. We had sort of one last day, and then we have a project out there that you were supposed to be working on. I've just gotten the word that as long as you are currently active in the classroom, I'm not in as much a hurry to compile a final grade for you. So that's good news. That means that you have a little extra time on that project. We don't have to work on getting that right away. Here's a fun little biology project I started today. These are little extenders I printed on the 3D printer. So these are going to go to the hospital. I have a cousin who's working in the hospital right now. And these just go all across the back of their head, and it allows them to take the elastics off of their ears and put them over these little hooks. So that's a fun little project that I started yesterday, and yeah, I'm excited to see where that goes. She's currently in self-isolation right now because she was working closely with the COVID, so anyways. We are going to be starting the unit on genetics today. It's formerly Unit 1. I guess I should update that. Unit 3. Okay. I'm still debating about if I like that this new unit or whatever, but I'm also noticing that most of you have already signed up for the Google Hangout. So as soon as I have the majority of the students in that Hangout, we'll actually hold a class in there using a live stream, probably or using a live video chat at some point. So that's my hope, at least. We'll schedule that at a later date. I have had a couple of slowdowns today in the stream, so just hang through. We did get through the whole class earlier, so we were able to do it. It was just there was a couple of lags, and I can already see that my kilobytes per second is dropping, so not sure why that is, but we'll CPU. Maybe my computer is just chugging. This thing is using up a lot of the life in my computer here, so we'll see. We can drop a couple of things like that. Yeah. Good. Okay. Well, definitely a couple things that are using up some of my data here. Okay. Interesting. Okay. I am going to close that at least. I can close this one. Okay. Get rid of some of this extra. All right, let's get started. So we are going to get started today on genetics. I'm very excited. Genetics is a very fun unit. It is a unit that some of you, if you remember grade 9 science, there was a genetics unit in grade 9 science that has a lot of the same material. Depending on your teacher, they might have even covered the majority of this material. So it sometimes is a very straightforward unit for some students. Other students, it's been a while since you were in grade 9, and that's cool too, because we're going to cover it all from the beginning. So some things that we know already, every living organism is made up of cells, and these cells express many different traits or distinguishing characteristics. These distinguishing characteristics are essential to separating two different individual cells from each other. They're also essential to separating two different species of cells apart. When we talk about the transmission, the movement of traits from one species to another, for one organism to the next, this is a concept called heredity. So genetics is the study is essentially the study is, does not want to let me type here, okay, is the study of the patterns of inheritance of traits over multiple generations. That's sort of the definition I might use. So cell theory tells us that all cells come from other cells. So by the principle of inheritance, all cells inherit their traits from their parent cells, and those parent cells from the previous cells, and those from the previous. And this is one of the major concepts that is also supports our evolutionary theory, right? That since everything comes from the past, then all the traits that organisms carry today are not actually any different than that are the same as the traits that are carried that were from an older generation because of mutation. So there's a cell cycle that we can look at now. This is the process for any cell that is going to reproduce, and so this process is Whoa, okay. Looks like I'm still going. Okay, that's better. All right, so the cell cycle is the process of growth and birth, I guess, in cells. So cells reproduce by a cycle of growing and dividing called the cell cycle. One cycle is where one cell will become two. This is the cell cycle of mitosis. Mitosis. Okay. So in mitosis, a cell grows throughout the interphase stage. This is where you can see growth is happening in all stages of interphase, and then it undergoes mitosis. I guess mitosis is really this phase. So this is our mitosis phase. This is our interphase or growth phase, and then our mitosis or division phase. And so the cell undergoes a process where we have prophase, then metaphase, then anaphase, then telophase or telophase, and finally cytokinesis, and one cell will become two. This is a process that happens because when cells become too large, their efficiency for eating and for absorption of nutrients drops. So as an object's volume increases, the surface area decreases. And since cells depend on their surface area for food comes in through the surface area, through the cell wall. Then as the cell size increases, then the amount of exchange that can happen decreases. Okay, just trying to investigate if there's some culprit behind this sudden network drop, but cannot see anything particularly rambunctious in my system here. Okay, one day we'll figure it out. So when a cell, this is because of the volume ratio. So when the cell is small cell to a small volume. So when it's small, let's just say this. So when it's small, the volume to surface area ratio is similar. You might even have a high surface area to low volume. So this is high and this is small. When the cell becomes medium, then the volume and surface area are more similar. And when the cell becomes large, then the volume is large, but the surface area is gone down. So the ratio of surface area to volume has altered. This is bad news because cells eat through their surface area. The food enters the cell. Food enters cell through the cell membrane. So large cells need more food, but they have lower ratio of surface area. So less food gets in. And so then the cell becomes starved if it gets too big. So when cells are getting so big, at a certain point, the cell actually can start to starve itself. And at that point, that sets up a chain link of events that causes the process of mitosis to start to kick in. And the cells will suddenly start to engage in this process, try to divide themselves, and through this division, create two new cells. So here is an example of what that looks like. This is the process up here called mitosis. So this is mitosis. And this is what we call meiosis. It's also a very important process, but this process only applies to, or mostly applies to sex cells. It also applies to some plants that undergo meiosis to form certain structures or certain generations of the plant, but and certain animals that also have that process for certain phases of their lives. But mostly we are talking about mitosis for growth. This one's for growth, and this process is for reproduction. So two types of reproduction can occur. We can have asexual reproduction. This is when results when a single celled organism splits in half. There are some examples of animals that asexually reproduce, like through fragmentation. A sponge, we know, can asexually reproduce. Excuse me. It seems odd to do these sexual reproduction discussion now when we've been talking about it already for several months. And of course, there's also sexual reproduction, which is the unification of two gametes, which is a sperm and an egg, typically, or the equivalent in other organisms, which are created by meiosis. So somatic cells, these are called our body cells, or non-sexual cells, are somatic cells, and many asexual organisms also use mitosis as the process for their reproduction. So this has the two phases, the growth phase and then the division phase. This is so the growth phase is just where the cell grows, it gets bigger and bigger, and then the mitosis is that division phase where the cell divides into two. The new cells are produced for growth and repair. They are genetically identical, or as close as we can get, to the previously existing cells. So this is a key point about mitosis. So mitosis creates mitosis. We're just going to say this over and over again for the next month. Mitosis creates two genetically identical daughter cells. Okay, so that's a key point. We gotta keep that in mind. So mitosis, the instructions that tell each cell how to form a new cell are stored in the DNA in the nucleus. This is important because that DNA will inform how to make those cells different from each other, different from other organisms of the same species, or even different from other species, because they are always pulling that information from the DNA. The DNA is contained within a form called chromosomes. Chromosomes are complicated. They are a mixture. This is a form, so this is a combination of protein and DNA molecules. And this combination of protein and DNA molecules are kind of wrapped in on each other, and they form these sort of structures. They're large enough that they can be seen in using the right kind of microscope. So whether that's that might be electron microscope, I believe is appropriate for seeing these. They are quite small. So that's a chromosome. A chromosome is one strand of DNA wrapped around many different proteins. Those proteins are called histones, is another word for those proteins. So if you're ever interested in researching into that, those proteins are known as histones, histone proteins. And of proteins and DNA molecule. So it's one DNA molecule, sort of thought of as like a big long thing, and then that big long strand is wrapped around these sort of histones that are kind of, located at different spots, and then it might even like wrap around itself. So here we have the DNA. We'll talk about this more in our next unit, and these are the histones. And then all of this together, if we zoom out, zoom out, and it looks like just sort of a big mess, but it's kind of all crumpled together, and we would have to kind of imagine, I don't know, like little, oops, too big, little, there are little green dots all over it, but we can't really see those so much, but they are in there, little green dots of the histones and and the DNA molecule. And so those histones are really there to help hold everything together and make it into this sort of one bigger unit. So a body cell, a somatic cell, so your skin cells or your white blood cells or your eye cells, all those cells contain 2 copies of every chromosome. Each chromosome in the human body, there are 23 of them, is a different length, and it contains different information on it. So there are 23 types of chromosomes. So humans on average carry 23 types of chromosomes, 23 different types of chromosomes, numbered according to their size. So each of the 23 chromosomes is a little bit different in terms of size. There's a really big one, it's number 1, and then there's a little bit of a shorter one, it's number 2, and then number 3 is a little bit shorter than that. And so number 22 is a really tiny little chromosome. And we carry 23 different types in a diploid cell, like a skin cell. There are 2 copies of each type. And as we'll soon learn in like a matter of seconds, there is the reason we have 2 copies is that one of those copies came from your mom, one of those copies came from your dad or your equivalent donors. One of them came from the sperm, one of them came from the egg that were formed together to form your zygote. So in mitosis, I do want to do a little quick recap of mitosis. This is a process that you might have gone through in a different course at some point, but we are going to go through the 4 stages of mitosis real brief so that we can talk about meiosis, which is the stage that is important to heredity. So mitosis is this 4 stages of the cell. It's not really a focus of the course. I don't usually even do that many assignments on it or anything. I just want to get you to make sure you know it very well because it is something that shows up in biology quite frequently. So we have the prophase, the metaphase, the anaphase, and the telophase. As I always call it telophase, but I guess apparently it's telophase. So interphase is the phase before mitosis. It is a phase where the cell grows. During that growth phase, there is a point where the DNA and chromosomes are in a loose form. And so this is called chromatin. That chromatin, the histones are not holding on to the DNA very tightly. So in chromatin, the histones have relaxed their hold on the DNA so that it is loose and easily read by other proteins in the nucleus. And as we'll mention on the next page, just because while we're here, in later interphase, the histones will wrap themselves more tightly in DNA, and the strands will become compact chromatids. So here is the chromosome. This is an early, this would be sort of early interphase here. So this is early interphase. And you can see the chromatin is all dispersed. The chromosomes are very loose. Chromosomes here are just sort of numbered, just a name for this DNA, this unit of DNA. So in mid or late interphase, this chromatin will actually duplicate, and after duplicate, the pairs of the chromatin will coil up, forming the chromatids. These chromatids, they're not called a tetrad. Please erase this as an old mistake that keeps showing up. Forming a pair of chromatids. These are known as the sister chromatids. These are sister chromatids. Tetrads is another thing that shows up a little later. So please make sure you correct that in your booklet. And I'm going to make a note, and hopefully this time I'll correct it for sure. Okay. I said every year for the last, 4 or 5 years. Okay. So before mitosis, each chromosome is formed of a pair of identical chromatids connected together by a centromer. So this pair, or the sister chromatids, they are connected together by a centromere. So you can see here, this is the chromosome before mitosis. It's already been condensed. This is not really how it is, but this would be called one chromatid, and it's one chromatid in this one chromosome. This is now in late interphase. We have one chromosome, but that chromosome is made of the two chromatids. So these are the sister chromatids. And these are identical sister chromatids held together by a centromer. At the end of mitosis, we're going to be back at a state where we have one chromatid per chromosome. Notice that in all cases, this has been a single chromosome. So we still call all three examples here one chromosome, even if it is made of multiple chromatids. And that's a tricky one that I find that some students get frustrated with or confused by. I'll put a sad face in there. So do watch out for that. I'm going to just make this something colored. So we still call them, we would still call all three examples here chromosomes, one chromosome, even if it's made of multiple chromatids. So you might have to count at one point, count how many chromosomes, how many chromatids, etc. So at the beginning of prophase, then we have a bunch of chromosomes. All of those chromosomes are made of chromatids. I'm looking at this. This looks like it's a picture of. Hey, what's this doing here? This is meiosis. That's meiosis. Get rid of here. Get out of here, meiosis. Okay, what are you doing in there? Let me see if I can drop an example in. Whoops. See if I can. How quick can I find prophase? Oh, it is exactly the picture that comes up when you search prophase. But I have to pull the second picture. There we go. Okay. Whoops. Okay, so that's prophase 1. I wonder if I can just. Oh yeah, for me this is easy. Okay, so I'm gonna do that. I'm gonna cheat because I can do that. That was an old picture of meiosis. Watch out. I'm gonna replace that one with this new picture from mitosis. Boy, a couple mistakes in this booklet. I am grabbing my pencil right now. I'm making 2 notes right here. So there's that prophase picture, and then the tetrads. Leftovers from a time where I didn't know as much. It takes a while to learn this stuff. Okay, so during prophase, the nuclear envelope disintegrates. It starts to break down, consumed for its nutrients, and then there are these things called the centromeres, or the centrosomes. These are the centrosomes here. They start to form the microtubules, which are these little lines that are in between. And those are going to be used to drag stuff around the cell. Now that the nucleus is gone, the chromosomes are exposed to the cytoplasm. This is one of the reasons that the chromosomes have to be in the condensed chromatid form, because this protects the DNA in the cytoplasm. So the DNA is now exposed to the dangers of the cytoplasm. So they need to stay in the chromatid form, which is more durable. So we have these chromosomes. They now start to align themselves. This is called metaphase because it's in the middle. Meta is, mid and meta. They all have sort of terms. So they are aligned along the middle. This is called the metaphase plate. This alignment is assisted by the spindle fibers, which come from the centrosomes. Those attach themselves to those centromers, these little center protein packs that are right in the center of the sister chromatids, and then the centromer will actually divide itself during anaphase. As the centromer divides, the sister chromatids are separated and move to either side of the cell. During telophase, the cells become even farther apart. The two sides of the cell become farther and farther apart from each other, and there starts to form what is known as a cleavage. This is the cleavage. How do you spell cleavage? Cleavage site. Cleavage line here, or whatever. And then the cell will divide, and that's under a process known as cytokinesis. Kinesis is action or movement sometimes, and you're splicing the the cell environment in half. So the result is the result is that the chromosomes that were formed of two chromatids have been separated so that each of the daughter cells contains an equal number of chromosomes. These, where each chromosome is back to its original one chromatid configuration. So one way I like to think about it is that I think about it as you have at an interphase beginning. You have one set of data. Then there is a duplicating. So then you have a late interphase. You have two sets of data. And then at the end of mitosis, you have two cells with one set of data each. And that's mitosis. So comparing this to meiosis, we are going to see some differences happening here. So here's the setup. You have two chromosomes. These chromosomes are then organized in prophase. The prophase later in metaphase, they are lined up. They are divided through anaphase, and then telophase and cytokinesis. We get a division of the cell itself, and we are left with two identical daughter cells, each one with half the number of data, half the number of chromatids, but the same amount of data as what we started with. So same number of chromosomes, but same number of chromosomes. So that's probably the most important part. So that's mitosis, same number of chromosomes as earlier. So we call this 2N for diploid because we use the word 2N. This is a diploid. It means 2 of each chromosome type. And those types are numbered, so that's why we use N for number. So remember that we have that humans have 23 chromosomes, chromosomes 1 through 22, and then X and Y, which are the same sex type chromosome. And so then the diploid, so 2N equals 2 times 23, which equals 46 chromosomes in a diploid cell. Okay, so let's compare this to meiosis. This is how we form haploid cells or sperm and egg cells. So meiosis produces reproductive cells, the sperm or egg. They contain only one copy of each type of chromosome. This is important because when we combine the sperm and the egg, we will now have a full set of 46. So if we take two cells that each have 23 chromosomes and we combine them, we'll be left with a new cell that has 46 chromosomes. On average, they contain only one copy of each chromosome. There are examples later on in our unit where we will have some examples that break this this trend, but this is what we're going to go with for now. The gametes are the egg and sperm, and they are the ones that are going to contain 22 autosomes and one sex chromosome on average. There are definitely lots of different examples where the gametes contain different combinations of these sex and autosome chromosomes, and we'll talk about those later in a different section. So the phases of meiosis are the same phases. We have interphase, where the chromosomes will be replicated to make chromosomes with two chromatids. So we'll double the chromatids. So this is what's going to happen in interphase. And then phase 1 of meiosis is going to be the first phase where we actually divide away the number of chromosomes. And the first phase is simply called meiosis 1. It consists of prophase 1, metaphase 1, anaphase 1, and telophase 1 or telophase 1. And then we're going to have a meiosis 2 where we're going to cut down the number of chromatids. So this is going to cut chromosomes. Divide number of chromosomes. And then this one here is going to divide number of chromatids. Okay, so let's give this a look. So in prophase 1, we are going to have a diploid cell. Each diploid cell has the 2 copies of every chromosome. Each of those chromosomes carries carrying 2 chromatids from the original interphase. And all chromosomes have the 2 sister chromatids. So they are all in this format that looks like this. This is the format. So we have 2 sisters, 2 sisters. Each pair of the chromosomes carrying the same genes, those homologous chromosomes, will become aligned in prophase 1. This is a little different from the previous. So these homologous chromosomes now, this is the name we're going to give them, are going to become aligned. So these are 2 chromosomes that have the same number, or in this case, same size is another way of looking at it. They're going to become aligned. So this would be, so both are same type. In this example, 1. They could both be chromosome 1. Why not? They could both be chromosome 2. It doesn't matter. So both of these chromosomes are going to align themselves together, and they will carry similar but not identical information. They might carry different forms of genes. This is what we call alleles. Different organizations, different mutations, all sorts of things. And they carry slightly different pieces of information on each. And so then something really important happens. This is the most important part of the whole thing. This is super, super important. This is what makes life really exciting for creatures that engage in sexual reproduction, is crossover. So during crossover, these non-sister chromatids, these homologous chromatids, exchange segments. So they actually will exchange pieces of each other, and that recombines the genes so that the chromatids are no longer purely one type of chromatid. They are now a mixture of the 2 chromosomes that you had before. And the result is that all 4 chromatids for that 1 chromosome are now all unique. So every chromatid is going to be unique after this crossover. Every chromatid is made unique by the crossover. And the other thing that's by the crossover, and the other thing that's really interesting is that not only are they unique, but they're unique in a random way. So we should actually say by the random crossover. And this is a big thing about our evolution unit that we ended up talking about, is how there's an influence of randomness in everything. So we can think of this as like an agent of genetic drift, is that crossover itself is a random event. So there's this random sort of shuffling of DNA that happens before even the sperm and egg are made, that's really at the core of all inheritance. So at this point, then the chromosomes align, but they are sort of stuck together. And then while they're stuck together like this, we call this a tetrad. So this is where the tetrad is. This is known as a tetrad. And it is when tetrad. This is the state point in meiosis where all four homologous, I'm going to say homologous because they are of the same type, homologous chromatids are stuck together. Okay, so after this point, then anaphase is the same sort of divisional phase, except instead of ripping the chromatids apart from one another in one chromosome, they are going to rip the chromosomes away from each other. And in this way, they end up doing something really important. They reduce the total number of chromosomes down by half. So the sister chromatids are held together. So one from each pair. So I'm going to be a little bit clear here that the sister chromatids, the tetrad is the tetrad, the two chromosomes, the two homologous chromosomes are torn away from each other, or is divided. Let's go with divided. Divided, leaving each side of the cell with half the original number of chromosomes. Each chromosome is still made up of two sister chromatids. So this can be a little bit hard to wrap your mind around, especially because the chromosomes, chromatids naming convention is very difficult to sort of wrap your mind around. But if you can get it, it'll make sense. But so here we have the sister chromatids here, right? Two sisters chromatids. Now no longer identical. And we have here now only two chromosomes. And two chromosomes. And we should note that in this example, there were originally 4 chromosomes. So originally we had 4 chromosomes. We've gone down to half. That means that we've gone from a diploid cell to a haploid cell. So then telophase happens. The process, it extends, it divides, and we now have 2 cells that each have only 2 chromosomes. Carry this through to another meiosis event, meiosis 2, and essentially the same thing happens again, except now each cell only has half as many chromosomes. Each of those chromosomes made of 2 chromatids. Those chromatids get organized in a meiosis, they get, in a metaphase, they get divided away in an anaphase, and we are left with 4 cells. Each of those cells has 2 chromosomes, and each of those cells, and so let's just put a little example here. Remember our original cell, and this original cell would have had, 2 copies of every chromosome. So let's make one blue, one red, like this, and let's make another one that's a little bit smaller, right? So this is our original, and it had 4 chromosomes. And now our haploid cell each has 2 chromosomes. And over here we have, notice that each of these has 2 chromosomes. So we've already lost half the information before before meiosis 2 even happens. So that's really an important key point there. Now, on a sort of unrelated note, we have cancers, and I think we can talk about cancer as being an uncontrolled growth. So most cells in your body have a known endpoint. They can only be divided a certain number of times, or they'll have a slow division rate that's required for that body part. But in cancers, one of the ways that cancers affects us is that the cells have an uncontrolled rate. And so these can end up crowding out tissues. That results in function loss. They could also interfere with other processes, and they can even pass this trait on to other cell types, and that can cause the cancer to spread. So the causes of these are things like mutagens, changes to the DNA, etc., from various factors. So here's the process. We have, in this example, we had 2 chromosomes. So here's our 2 chromosomes. So let's just think about this. This is 2. Those 2 DNAs are copied before prophase 1. Here's the crossover. This is crossover. I don't know why I can't spell crossover today. Crossover. And then with recombination and things. Then metaphase, the chromosomes line up. Anaphase, they are separated. As they separate, we now see that we have only 1 chromosome each, 1 chromosome. And then that 1 chromosome gets split up, and we are left with 1 chromosome each. It depends if you're counting out the diploid, the chromosomes or the chromatids in this case, as to whether you call it haploid, diploid, or tetraploid, but etc. So that's our little introduction. Tomorrow we'll talk about genetics in more detail with our discussion of Gregor Mendel in our next section. Please feel free to reach out to me in the Hangout. I'd love to talk to you there. And also, you can always send me an email or talk to me through the classroom. All right. Hope you have a great day, and we'll talk to you later.